So with the second lecture, we will start into the details of our photosystems. And so a photosystem consists of the pigments that we saw earlier, whether it be chlorophyll A, chlorophyll B, or accessory pigments. And so a photon of light strikes these pigments, and when the pigments are struck by that photon of light, they are excited, and the excitement of the pigment's electrons move those electrons into a higher state, so a different shell of the electron, and they bounce from one to the other. You can think of it like Newton's cradle, where when you strike one, moves to the next, moves to the next, moves to the next, until it comes off the other end and hits a special pair of chlorophyll A molecules. And these chlorophyll A molecules are part of our reaction center complex. And so this special pair of chlorophyll A molecules are our primary electron acceptors. They transfer the excited electrons up to the primary electron acceptor. And this redox reaction is one of the first steps in our light reactions. And so we have two photosystems within the light-dependent reactions. The first of these photosystems is photosystem 2, and the second you will see is photosystem 1. And they are named for the order in which they were discovered, and nothing more or nothing less. So the beginning of our photosynthesis and our light reactions is photosystem 2. And the wavelength of light that excites our special chlorophyll A molecules in here is a 680 nanometer photon of light. And so these two chlorophyll A molecules are best at absorbing 680 nanometer wavelength light. Once the photon excites the pigment molecules' electrons, then they end up in this reaction center. The electrons are excited, water comes in, is split, it donates its 2 electrons. Oxygen is released and it combines into O2. And the hydrogens are released into the thylakoid space. Then the excited electrons move up into this electron transport chain. So you have the excited electrons from the pigment molecule that were excited by the photon of light. You have the electrons released from the water. These are all moved into a higher state, a higher energy state. And then they move through this electron transport chain and generate ATP. And we'll see a little more detail on this in a few minutes. So photosystem 2 is responsible for making the ATP. And so the electrons that move through this electron transport chain now enter into photosystem 1. Notice they are still at a slightly higher excitation state than they were when they entered the P680 over here, the photosystem 2. And so they're a little bit higher energy state than they were in photosystem 2. Another photon of light strikes, and these chlorophyll A molecules like to absorb 700 nanometer wavelengths. This excites them to an even higher state of excitation in photosystem 1. And then they move through another electron transport chain, a series of proteins. Ferredoxin is the first that it hits, and then it hits NADP reductase. And so NADP reductase is going to add the electrons and a hydrogen onto NADP plus. It's going to reduce NADP plus. And now NADPH will be our electron carrier along with the hydrogen. So you can think of this as a group of workers. Our photon of light hits the light pigments, excites the electrons in photosystem 2, moves those electrons to a higher energy state. Then they go through the first electron transport chain between photosystem 2 and photosystem 1. They release some of that energy to pump protons in, so you had the hydrogens released from the water into the thylakoid space, and then these guys are fueling an electron pump, a proton pump, not an electron pump, a proton pump. The electrons are fueling a proton pump and pumping 4 more molecules of hydrogen in to build the concentration gradient. And then those hydrogens run through ATP synthase to make ATP, and we'll see that in the upcoming slides. Electrons are at a higher excitation state because they came in over here with photosystem 2 and they're entering photosystem 1 all the way up here. Another photon of light, the 700 nanometer light, excites them to an even higher state where they go through another electron transport chain and reduce NADP into NADPH. So the products of photosystem 2 and 1 are ATP and NADPH, which are then carried, as we saw before, over into the Calvin cycle to make glucose. So again, our mitochondria that are doing cellular respiration are chloroplast, responsible for photosynthesis. Both have a similar makeup. They have a membrane that the electron transport chain is embedded in. The hydrogen in the mitochondria is pumped from the matrix into the intermembrane space. The hydrogens in the chloroplast are transported from the stroma, which is that fluid surrounding the thylakoids, into the thylakoid space, creates that hydrogen gradient, and then it runs back through ATP synthase to generate ATP. So mitochondria, chloroplast, very similar makeup. Use electron transport chains to build up hydrogen gradients and then run those hydrogen gradients through passive diffusion back through ATP synthase to make ATP. And keep in mind, these are all called proton pumps because hydrogen gave away its electrons, and hydrogen's basically made up of 1 proton and 1 electron. And so when it gives away its electron, it is basically just a proton. So that's why you'll see these proton pumps pumping H+, which is basically just a hydrogen molecule with this 1 proton left. And so here you can really visualize our light reactions. The energy from a photon of light excites photosystem 2, and in particular these guys like 680 nanometers of light excite the electrons in that pigment. Also have the splitting of water that donates electrons into the system. The oxygen is released as a byproduct and forms oxygen, O2, which then moves out of the cell to be released for us to breathe and to carry out cellular respiration. So the excited electrons run through this electron transport chain. So you have the 2 hydrogens released from water, and then you pump more hydrogens in from the stroma into this thylakoid space. Another 4 hydrogens build up that concentration gradient, and then the hydrogens both from water and from the proton pump, fueled by the electron transport chain, now run through ATP synthase back out into the stroma. And that hydrogen running through there through passive diffusion generates ATP. That ATP then goes into the Calvin cycle. the excited electrons that have ran through this electron transport chain and fueled this proton pump now move into photosystem 1 at a slightly higher excitation state, struck by another photon of light. 700 nanometers of light is what wavelength of light is what photosystem 1 likes to work at, excites the electrons to a higher energy state. They move through another electron transport chain and fuel ADP reductase to reduce NADP to NADPH. Now NADPH, our electron carrier, also moves into the Calvin cycle. It's going to be our reducing agent in our Calvin cycle, and ATP will provide the energy. And so you have a Bioflix animation you can find in the study area of Blackboard, where you can watch the animation on the light reactions. So be sure to pause here and go watch that real quick in your study area. Okay, so we have seen what happens with our light reactions. Products of the light reaction are the oxygen, ATP, NADPH. Those move into the Calvin cycle along with carbon dioxide to run the Calvin cycle and make a carbohydrate in the form of sugar. So the Calvin cycle is broken into 3 different paths. Phase 1 is our carbon fixation. And so this is going to show 3 cycles of the Calvin cycle, because we have 3 CO2s coming in, and with each CO2 entering, we have 1 cycle. So CO2, each CO2 runs 1 cycle of the Calvin cycle. And so the CO2 forms a 6-carbon molecule by combining with a 5-carbon sugar. So ribose, over here, ribulose bisphosphate, RUBP, joins with 1 carbon of the carbon dioxide using the enzyme rubisco. And it's thought that rubisco is one of the most plentiful proteins on planet Earth. This catalyzes that formation of a 6-carbon molecule, which is very short-lived, very unstable, and immediately breaks into 2 3-carbon molecules. Those 2 3-carbon molecules now move into the reduction phase. And so with the reduction phase, that 3-carbon molecule is reduced to the product that eventually forms glucose, G3P. And G3P is glyceraldehyde 3-phosphate. And so 1 molecule, so you've made 6 molecules of G3P. 1 is released out as output, and then the other 5 remain in the cycle to be recycled. So regeneration, phase 3 of the Calvin cycle, is regeneration of the CO2 acceptor, ribulose bisphosphate, and so these 5 come back together to make 3 molecules of ribulose bisphosphate. I have a hard time saying that there. Sorry. And then start the cycle all over again. And so phase 1 is carbon fixation, fixing the carbon from CO2 onto the 5-carbon molecule ribulose bisphosphate. 6-carbon immediately breaks into 3, then ATP, NADPH fuel the reduction to glyceraldehyde 3-phosphate. 1 of the molecules made leave the Calvin cycle, the other 5 regenerated to carry on the cycle. And so after 6 total turns of this cycle, you get 1 carbon for each cycle, and you need 6 cycles, 6 carbons to make glucose. You end up with a new molecule of glucose. And so again, you have a animation of the Calvin cycle in the study area of Blackboard. Go look at it real quick before you continue on.